Gold nanoparticles may be used to deliver a drug to a targeted area, like a molecular capsule, in a controlled release manner before eventually being broken down by the cell. Nanoparticles are submicronic colloidal systems which are able to withstand onconic pressure exerted by proteins to pull water into the circulatory system. Nanoparticles must have the ability to flow freely in fluid without aggregating or exhibiting off-target effects. Nanoparticles are beneficial due to their biological compatibility, optical properties, as well as its greater stability due to gold-sulfur bonds. Nanoparticles are small enough to travel around the body and into cells without trouble. They are also large enough to carry therapeutics to treat disease. Nanoparticles are actually larger than a small molecule. They can vary in size, with some nanoparticles being 1 nanometer in diameter, while others are 2,500 nanometers. Nanoparticles act much like the vesicle coat proteins and adapter proteins used for vesicle trafficking specification. A drug being carried by nanoparticles is similar to the molecular cargo that vesicles carry. Nanoparticles have the ability to enter a cell through endocytosis, in which the nanoparticles itself is the cargo being carried into the cell by a vesicle. Nanoparticles can be differentiated by assembly, surface modification such as clathrin, size, and shape. These varieties provide flexibility in function and specificity. Nanoparticles can also enter the cell by direct diffusion or ion channels. They are often biocompatible, as polymer engineering offers a wide range of possibilities for their composition. This is a major reason why they are helpful in aiding the passage of drugs into certain cells. They also have a larger surface to mass ratio than most particles, allowing them to bind and carry drugs. Their size allows them to cross the biological barriers of the body while also being small enough to reach even the nucleus of a cell. Depicted here is an electron microscopic image of HIV fusing with the plasma membrane of a cell. HIV binds to the target cell surface receptor based on its vesicle coat properties. HIV then fuses with the plasma membrane, which allows the viral nucleic acid inside the nucleocapsid to enter the cytosol, where it replicates. Envelope glycoprotein GP120 is a protein expressed on the surface of the HIV envelope and is a potential target of certain nanoparticle treatments for HIV. It allows the virus to enter the cell by attaching to the cell surface receptors DC sign, heparin sulfate proteoglycan, and CD4. Because of this, inhibiting it could likely prevent HIV's access to the host cell. When the protein binds to CD4, it causes conformational changes in certain G proteins that cause the HIV virus to fuse to the cell and infect it with viral DNA. The potency was enhanced for both viral infection inhibition, cell-free virus breakdown, and inhibiting HIV-1's ability to replicate. Nanoparticles are able to cross the blood-brain barrier and therefore deliver drugs. Nanoparticles have a variety of applications. These applications include, but are not limited to, antimicrobials, the cosmetic industry, photothermal therapies, photoimaging, nucleic acid delivery, and treatments against cancer. The future of nanoparticle research has an almost unlimited amount of possibilities of material to use and treatments to provide. Scientists are trying to develop single nanoparticles that will deliver multiple treatments for different illnesses with only one injection. Some of the main research for nanoparticles is in the field of cancer treatment. Though many cancer treatments are being created, lung cancer is being specifically focused on. Nanoparticles are currently being made out of artificial macromolecules.